Well, thanks for letting me talk today. It's interesting being called a new biotech, which we certainly are, but we didn't appear overnight from no work. The work I'm going to talk to you about today is probably the result of around 15 years of research on stem cells and over 30 years of work on spinal cord injury by our co-founder, Michael Feelings. The cost to get to this point is around $15 to $17 million in academic funding, and except for all the funders were already mentioned. So it's really the genesis of a lot of time, a lot of dollars, and a lot of hours to get here. So we are a company which is primarily using stem cell therapy for the treatment of acute and chronic spinal cord injury. We are exclusively using human-induced pluripotent stem cells, iPSCs, and our goal is to restore lost function and independence for people living with traumatic spinal cord injury. So what I'm going to show you is some data today, and it refers exclusively to traumatic cervical spinal cord injury. And we're currently raising, as I guess every other company is doing here, to advance our technology and hopefully interaction with the FDA. As I mentioned, we've been academic lab has been around for several years, and because of that, we've generated quite a large IP portfolio. We're based at UHN, which is the largest research hospital in Canada, and Toronto Western Hospital specifically. And because of this, we've now licensed in a World Life exclusive license for all our technologies from, do I have a pointer anywhere on this one? How does the pointer work? Oops, shit. Okay, I can't see it. But anyway, the key IP that I'm going to talk today about is the generation of oligodendrogenic neuroprogenitor cells, and we've just had a worldwide patent granted for that, and also methods for generating neuroprogenitor cells with spinal cord identity. So as people have alluded to prior, what are you actually targeting and what are you hoping to treat? Well, for us, after interactions with patients, it really is restoration of bowel and bladder function and restoration of hand and arm function. So if you can see here, this person really doesn't have the power grip or the precision grip to hold a cup, feed themselves, dress themselves. So these are some of the key metrics that we're looking on to try and restore. And we think we've done it, in an animal model at least. So here we have two rats, the one on the left using conventional MPCs, and by that I mean forebrain identity MPCs. You can see that at 12 weeks after injury, she's unable to grasp that food pellet, and she can't feed herself, she can't do anything really. The rat on the right has been treated with our restart repair cells, and here you can see, and this is 12 weeks post-injury, of a C6, C7 cervical injury. She's now got that ability, that precision and power grip to grasp that food pellet and feed herself. So these are the metrics that we've been using internally in the lab to determine if our results, our research is being successful. So we're strong believers in cell loss is the underlying cause of pathology in spinal cord injury, but also a host of other central nervous systems disease, such as stroke, traumatic brain injury, multiple sclerosis, and of course, traumatic spinal cord injury. And it's the loss not just of the neurons, but the oligodendrocytes and the astrocytes. So really to have a chance of getting a therapy that will translate into functional recovery in patients, we feel cell replacement therapy is probably the optimal treatment therapy from our point of view. And here we are focusing on replacing all of the cell types that have been lost through the injury. So we maintain tripotency, so we put oligodendrocytes back in, neurons back in, and astrocytes back in. So when we've looked at our own research and research of others and clinical trials in the past, we've identified 3 critical limitations in current stem cell therapies for spinal cord injury. Now, the first one is that stem cells often lack regional identity, making them less effective. In acute and subacute injury, there's been a lack of focus on remyelination and myelination by the transplanted stem cells. And for chronic spinal cord injury, It's the most difficult condition that you can really treat with just the harsh microenvironment that you're dealing with. So you really have to tailor the therapy to the pathophysiology of the condition. And I think the way we've done this at Inteligex is we have looked at the pathobiology of disease, and we've tried to develop therapies specifically for the type of injury and the type of conditions that we see in spinal cord injury. So what we've done is we've generated a cell platform called the Restart Cell Platform, which stands for Region-Specific Stem Cell and Regenerative Therapeutic. I didn't come up with that. But basically, what we do is we take a human GMP iPSC cell line, and at the moment, we're working on allogenic, so this will be off the shelf. However, it's our goal to move this forward to autologous stem cell therapy when that technology has advanced enough. We then differentiate our cells into neural precursor cells, and then this is where the magic starts happening, and we place on the cell's regional identity. So we make our NPCs either forebrain identity, cervical identity, or thoracic identity. We then modify the cells depending upon the stage of the condition, whether it's acute or subacute. For restart repair, we do concentrate on oligodendrocyte lineage biasing. And for chronic spinal cord injury, we have restart regenerate, which is more focused on neural regeneration. And I'll talk about each of these components individually. So the first one is optimal spinal cord regeneration requires cells with spinal cord identity. We have a lot of underlying basic research into this, which shows that through RNA sequence expression, the expression of the central nervous system is different in the forebrain, the brain, and the spinal cord. And indeed, within the spinal cord, the cervical spinal cord has different expression profiles than the thoracic cord. So this is how we produce our regional specific identity NPCs. Really, it's just a differentiation protocol. But I won't go into too much depth about that. But what we do is we've transplanted these cervical NPCs into our rodent model of spinal cord injury. It's a C6, C7 compression contusion form of injury. And when we do this, we can see that the cells maintain tripotency, and they still have the ratios of neurons to astrocytes to oligodendrocytes as we experience with NPCs, but it's really the types of cells which we're now producing, FOXP1 for spinal excitatory interneurons, and HB9 for motor neurons. The good thing about these cells is these exogenous cells integrate with endogenous neural networks, and we do get an increase in these formation of these synaptic integration above what we would get with forebrain identity. So it's showing that we're getting increased synapse formation compared to conventional NPCs. And of course, when we do functional studies, here we're using grip strength and inclined plane, we show a increase in functional recovery using these regional identity stem cells. Now we come on to moving away from the region, but looking at the pathobiology of disease for subacute and chronic spinal cord injury. And we really feel that for subacute, you have to concentrate on myelination and remyelination of the denuded axons. So again, here is our modification of our cells, our restart cells. And here we do oligodendrocyte lineage biasing. And when we bias these cells, what it basically means is we increase the ratio of oligodendrocytes, but we maintain tripotency. We're very much of the opinion that you have to have all constituent cells and parts of the CNS when you do this. So we increase the ratio of oligodendrocytes, we maintain the ratio of neurons, but we decrease a little bit the astrocyte component of the cells. And as you can see with APC, we're getting myelination of these cells. And of course, this is a slide that actually shows the myelination. When we look at electron transmission EM, we're seeing that we are getting exogenous stem cells, which are myelinating endogenous axons. Likewise, when we do functional readouts, again, with grip strength, we see an increase in functional recovery with MPCs, and these are just restart repair cells. But interestingly, we also see tissue preservation as well relative to the use of MPCs or vehicles. So we believe we're actually preserving a lot of the white and gray matter from cellular death. Now, for chronic spinal cord injury, this is where it gets really difficult. And you've got to do a much more complex approach, and you really have to understand the pathobiology of the condition that results from a chronic spinal cord injury. And here you have a shift in the cellular profile that you take. You move away from oligodendrocytes because you're no longer wishing to preserve and prevent the death of the axons. Unfortunately, the axons have already died and the neural pathways have been lost. So here you have to really concentrate on neuron regeneration and re-establishment of those neuronal pathways. So here we take a little different. We're no longer biasing ourselves with a differentiation protocol. Here what we do is we express two factors. One is CHABC, and the other is GDNF, or glial-derived neurotrophic factor. So if we look at CHABC, one of the key things we have to do if we're going to treat chronic spinal cord injury is we have to be able to degrade the glial scar and grow into the cavity. And this is a very, very simple illustration of this. Here we're showing CSPGs, which are a component of a glial scar. The green cells on the wild type are just conventional MPCs, which are expressing GFP protein. And you can see that they can't cross or degrade into the CSPG, so they can't transverse the cavity. Here with our cells expressing CHABC, we're now able to degrade that glial scar and grow across the cavity in vitro. What does this look like in vivo? And this is just a snapshot of quite a bit of data we've got on here. If we look at the injury epicenter, we've injected stem cells rostral and caudal to that injury epicenter, and we can see that we're getting long axonal growth across the cavity and across the injury epicenter that extend quite far down to T5 in this model. So we have the ability now to degrade the glial scar and cross the cavity. So now we've really got to focus on promote regeneration, and this we do with expression of GDNF. And GDNF is beautiful because this takes it away, and it's very similar to what I showed with the oligodendrocytes, is we change the ratio of the tripotency of the cells. Here we change from 20% neurons using control MPCs up to 35%, 34, 35% using GDNF expression. So we've moved the clock quite a bit to the right to really promote those neuronal producing cells, and which again, we hope will form neuronal networks. We maintain the oligodendrocyte proportion, so we are still remyelinating any new cells or any new neurons and axons that we generate. And of course, we also keep astrocytes to maintain that matrix that glues everything together in the regenerated cord. When we transplant these cells into a rodent model, again, we can see that we are generating neurons, and we are generating synaptic connections with these neurons, which is the key of what we've been trying to achieve. It's no good having neurons if they're not forming synaptic connections. And we have a lot of the electrophysiology data if people want to look at that, which confirms that these are actually transmitting signals across the injury epicenter. And of course, when we look at these cells with grip strength, inclined plane, we're getting positive results on functional recovery relative to a standard test. So where are we now? We have completed all our research with research-grade human-induced pluripotent cells. We've now optimized all our protocols with xeno-free cultures, so we're now ready to make the move from research grade to clinical GMP grade. We currently have 5 GMP clinical grade cell lines from 3 donors in our lab that are undergoing testing at the moment. We've got another 2 inbound in the next 1 month to 2 months. So really now we're working on what is going to be the basis of our final clinical product. And once we have that, we then do scale up and process development, and we've got a few companies that we've identified which actually can undertake that, and that it's no mean feat to find a company that can actually do that. And then we complete revalidation, safety, and tox in our rodent models, our porcine and our monkey models. And then hopefully we're going to get into a phase 1, phase 2 clinical trial and get it into people with a spinal cord injury. So this is our process. As everyone mentions, this is not cheap work. Stem cells are very, very expensive. Rodent models are very expensive. But I think we've identified a pathway now where we can really move this research forward and get it to the point where it needs to be, which is in the clinic. Okay.
Great, thank you for the presentation, Paul. I guess the first question I had for you is, it sounds like you have a number of products that you're developing for different areas of injury and then also different chronicity of injury. Which of these products or sub-products is most advanced? Or do you plan to bring all of the sub-products forward at the same time? How should we think about that?
So our lead indication is traumatic cervical spinal cord injury. If I go back to this slide, the beauty is they're all coming off this platform here with restart cells. So our first disease modality would be acute or subacute spinal cord injury. The reason for that is we only do differentiation in cell culture, which makes scale-up process development a lot easier than genetic engineering. As people have mentioned as well, there's also the pathway to the clinic with genetic engineering, CRISPR-Cas9, isn't as well defined as it is just for the simpler pathway. But everything we do for the restart repair can be transferred to restart regenerate. So it really is acute, subacute, spinal cord injury, cervical.
Got it, great. And then I was hoping you could just elaborate a little bit more on how you imagine the patient experience shaping up as you move towards the clinic. Do you plan to incorporate any immunosuppression with these allogeneic cells, and what kind of surgical procedure do you expect to use to implant the cells as well?
So the immunosuppression is a key issue, and we're just going into porcines right now. So we're transplanting our human stem cells into porcines right now. We personally don't believe we're going to need any fancy immune cloaking, although we do have those technologies lined up with key industry individuals at the moment. We want to avoid them if at all possible, because we think it brings a certain unknown uncertainty into the cells and also the moving forward with the FDA. So we're working on that now, and that's where Michael Fairlings comes in, because he's the clinician. He was the first surgeon in Canada to transplant cells from the Stem Cells Inc. clinical trial into a patient, so we do have a lot of experience with this. Those cells were well tolerated. The patient, I should say, tolerated the procedure quite well. At the moment, it would be a surgical procedure, rostral and caudral. However, we do have some collaborations with some other companies which we're looking at alternate ways of delivering the stem cells through exosome technology, and we've got projects going on, but I don't know how successful those will be. We do know surgical transplantation works.
Got it. Great, well, thank you so much for the presentation.
Excellent.
Really appreciate it.